Great. So I want to welcome everyone to today's Let's Talk MPTs webinar on developing live biotherapeutics for HIV prevention and MPTs. My name is Bethany Young-Holt, and I'm the director of the Initiative for MPTs, or the IMPT. We are a project of CAMI Health housed at the Public Health Institute. And I'm joined today by Cheryl Verbitsky, our program manager at the IMPT, and Kwon Woo, our intern. We have 80 people registered from across time zones joining us today. So as we wait for others to join our Zoom room, we invite you to type your name, your affiliation, and where you are located into the chat feature. The field of cervicovaginal microbiota is an exciting one that's been gaining a lot of momentum for its potential role in health and disease. And today's discussion on developing live biotherapeutics for HIV prevention and MPTs will feature presentations by Professor Anke Hemmerling, the director of the interdisciplinary MPH program at UC Berkeley School of Public Health, an associate adjunct professor in the UCSF Department of Obstetrics, Gynecology, and Reproductive Sciences, Professor Jacques Ravel, acting director at the Institute for Genome Sciences and professor of microbiology and immunology at the University of Maryland School of Medicine in Baltimore, and Professor Douglas Kwon, Director of Clinical Operations and Physician Scientist at the Ragon Institute, Mass General Hospital, MIT, and Harvard. If you have any questions for our speakers during their talks, please use the chat function in the chat feature in Zoom, and we will address them during the Q&A portion of the webinar. For technical issues, please chat with Kwon Woo, our tech host. Also, this webinar will be recorded, and the recording and related resources will be available on the IMPT website soon after the webinar. So some of you may recall that the first topic in our Let's Talk MPTs webinar series in December 2020 was on the role of the cervicovaginal microbiota on contraceptive and MPT development, where Jacques Ravel presented on the role of cervical vaginal microbiota on health and disease, methods for assessing vaginal microbiome, and more. If you're interested in that webinar, the recording is available on the IMPT website, as will this one. It'll be recorded and posted there soon after this webinar. And we're just so delighted to have Jacques back again by popular demand, along with Anke and Doug, to discuss advances in this exciting field. So for those who are joining this webinar series for the first time, and you may be new to the field of MPTs, multi-purpose prevention technologies, or MPTs, are products designed to simultaneously address HIV and other STIs and/or unintended pregnancy. The Initiative for MPTs was launched 15 years ago in 2009 as a product-neutral global platform of product developers, funders, policymakers, and advocates aimed to provide technical guidance and strategic planning related to MPTs. And over the past 15 years, we've brought together the voices and expertise of multidisciplinary stakeholders to identify field-wide priorities, gaps, and strategic actions to advance the field. We launched this webinar series, Let's Talk MPTs, in 2020 to engage experts from diverse disciplines across the globe to help address MPT and HIV prevention R&D priorities and gaps, and also to help foster networking and collaborations virtually and in person when we have the opportunity to meet in person. All past webinar recordings are available, like I said, on our website. We are, again, looking forward to an engaged and lively discussion today. Condoms are both internal and external are currently the only available MPT. New types of MPTs are in development that could revolutionize sexual and reproductive health by incorporating prevention of HIV and other STIs into contraceptives, which are less stigmatized. There are about 24 MPTs in development with active funding. Most of these are in early stages of development. Some are in clinical stages. As you can see on this slide, there are an array of delivery systems, targeted interventions, and targeted indications, rather, including unintended pregnancies, HIV, and other STIs. I want to thank Jim Turpin for this slide that he presented at a recent summit. One of the things that we do as the IMPT is to track the different MPTs in the pipeline. So if you're interested in seeing what is out there, you can check the database at mpts101.org. So the role of the vaginal microbiome for MPT development was highlighted in the opening remarks of Dr. Jeannie Marazzo, NIAID director at the MPT Summit last month in June, held at NIAID on the NIH campus. And I'm extremely grateful to NICHD and other NIH partners for sponsoring this summit, which convened 300 in-person and virtual participants from around the globe for 2 days, which really shows the momentum for the MPT field. The summit included 47 speakers covering 9 session topics, and featured presentations from NIH, HHS, WHO, and the White House leadership. So with that, I am really excited to introduce our featured speakers for this webinar. Dr. Anke Hemmerling is associate adjunct professor in the UCSF Department of Obstetrics and Gynecology and Reproductive Sciences at UCSF, and her research combines approaches in clinical medicine and public health with a focus on global women's health and STIs. And over the last 15 years, she's implemented several clinical trials in the US and South Africa, assessing live biotherapeutics, vaginal lactobacilli strains for the prevention of genital infections and STIs. Dr. Jacques Ravel is the acting director at the Institute for Genome Sciences and professor of microbiology and immunology at the University of Maryland School of Medicine in Baltimore, Maryland. He received his PhD degree from the University of Maryland College Park in environmental molecular microbiology and ecology, and performed his postdoc training at Johns Hopkins in the Department of Chemistry, working on natural product chemistry. And Douglas Kwon is a physician scientist at the Ragon Institute at Mass General Hospital, MIT, and Harvard, and is the director of the clinical operations at the Ragon Institute. He has a clinical practice in the Division of Infectious Disease at Mass General Hospital, and he received his MD, PhD degrees from New York University, and then underwent internal medicine training at UCSF and New York Hospital Medical Center. And then he completed his training at Mass General. So I am going to hand it over now to Anke. And as a reminder, questions will be addressed at the end of the presentation. So please do type your questions into the chat and we will then address them. Anke, over to you.
All right, let me see if the sharing works. Okay. Thanks, everyone. As Bethany already mentioned, at the recent MPT Summit in Washington, D.C., Jeannie Marazzo, the new director of NIAID, actually dedicated several minutes of her keynote speech to the vaginal microbiome and HIV risk. So this is a very timely webinar. Doug and I tried to work on our presentations to tell a little bit of a story. So we'll actually go around twice. I will start with an introduction to vaginal dysbiosis and development of LBPs. Jacques will talk a little bit about understanding the structure and functions of the vaginal microbiome. Doug will take over talking about mucosal immunity and tissue inflammation in HIV. And then we'll do a second round talking about recent trials that we conducted, and also will then end it with our take on what is coming next for the field. So for those of you that are not that familiar, just a few basics. The later talks will go into more detail here. The cervicovaginal microbiome changes during the lifespan. For reproductive age women, an optimal microbial community is dominated by lactobacillus that are acid-producing hydrogen peroxide lactic acid. And this acidic environment restricts the growth of pathogens. And as opposed to the gut microbiome in the vaginal space, bacterial diversity is a sign of dysbiosis. It's not a good thing. And dysbiosis is a disruption of the vaginal ecosystem that leads to an overgrowth of pathogens. So it shifts the whole space to a non-lactobacillus dominant state that can be transient or a longer episode. It is often asymptomatic, but can also have symptoms associated. For decades, we've been left to treat it with a number of antibiotics. Most of them lead within very few months to a recurrence in about a third of the women. In recent years, there have been a number of clinical studies trying to establish new treatment modalities for BVs, boric acid for biofilm disruption, antibiotic partner treatment or periodic treatment, topical estrogen, lactic acid, and also a number of live biotherapeutic products. There are a number of concepts around the etiology of BV recurrence. This is a slide I borrowed from Bradshaw and Brodmann. 3 big buckets. Either BV recurrence could be a failure to establish lactobacilli again after the antibiotics cleared the slate. It could be a failure to clear that biofilm of certain pathogens, or it could also be a reinfection from partners. We also learned about a number of factors that are impacting the vaginal microbiome, among them those here listed: geography, host genetic factors, hormonal fluctuations during the life course, but also during menses, exposure to semen, menstrual blood. And we also learned that BV is associated with increasing risk of a whole number of health conditions: HIV acquisition, HPV persistence, and cervical cancer, preterm labor, pelvic inflammatory disease, and urinary tract infection. The causal pathways are not that well established at this point. BV global prevalence and burden is significant. About 25% of women in any corner of the globe are affected. And the global burden just for treating symptomatic BV alone is $5 billion a year. And including all those sequelae, it's even much higher, up to $14 billion are the estimates. For a long time, the term live biotherapeutic products is newer. We called it probiotics for a long time. We're trying to distinguish us a little bit now from those probiotics that are already on the market, often mostly developed as food supplements. So they only need to meet FDA safety requirements, and they're not required to do efficacy trials. The problem is also that they're often non-human strains. They are often non-vaginal strains, gut strains, and often are orally administered, creating certain expectations for consumers that may or may not be met. And what's also problematic is that this easier path to marketing as GRAS products may diminish investors' interest in funding, large and expensive clinical trials of biotherapeutics that are actually aiming to be regulated as drugs. So this is definitely a problem for the field. A few years ago, Janneke van der Weiger did a meta-analysis. I think it was 20 or so studies at that time that she was able to include, reviewing clinical trials that have been conducted combining antibiotics and live biotherapeutic products for BV recurrence. A number of other authors also published papers, and across the board, the verdict was not very flattering: high heterogeneity, low study quality for outcome measurements and strain identification. And the issues were mainly in two buckets: study product issues around stability, administration regimen, gut strains, and often strain colonization, if it was measured at all, ceased basically shortly after study product. And there were also design issues. Those were often small studies with short follow-up periods that didn't measure colonization, or at least didn't measure its strain specifically, and also employed a whole array of different diagnostic criteria that were not streamlined at all. So really leading to a situation where we compared apples with oranges. What is also important to know that for now, we are mostly relying on two tests for BV. The AMSL criteria was developed in the early 80s. It's a point of care test often used in the clinic, where you check for 4 criteria: a higher vaginal pH, a positive whiff test with KOH, vaginal discharge, and then under a microscope, these clue cells that are coding mucosal cells. And if you have at least 3 of these criteria, you are diagnosed with BV. In the 90s, a lab-based test, a gram stain test, was introduced to basically evaluate under the microscope the presence or absence of lactobacilli, Gardnerella, and other curve bacteria. Again, a higher score would be diagnosed as BV, and a lower score characterized a healthy vaginal microbiome. So those are the tests that are really the building blocks of the current FDA guidance document that gets put out and renewed every 5 years or so for treatment of bacterial vaginosis. So this is very relevant for us. What is a little problematic is that it is developed as a test of cure of BV after treatment, and is not designed to determine BV recurrence. So, and you can see that the enrollment criteria, even what is determined to be BV at enrollment, are different. There's 4 AMSL criteria at enrollment. They only use 3 at the test of cure. Gram stain is mentioned as an enhancement, but really a secondary endpoint. So for us, in the space of trying to determine BV recurrence, there was, uncertainty what to do actually with those middle ground intermediate scores. Do they count for BV recurrence or not? Again, not leading to a streamlining of outcome measures across various studies, really also emphasizing that there is a big need for adding molecular methods. And here I borrowed an earlier slide from Jacques from a paper summarizing the findings of a paper from a few years ago that really illustrates that just using AMSL BV gets us really just the tip of the iceberg. If we use the lab-based Nugent test, it will get a little more of the iceberg. But in order to characterize the whole BV iceberg, we really need to include molecular diagnostics of BV and also work on the standardization of those tests. So plenty of challenges for the studies of vaginal dysbiosis and LBPs, in addition to the ones I already laid out. Is the fact that AMSL and Nugent scores does not help us to distinguish between causes for recurrent BV. Is it persistence, unsuccessful antibiotic treatment? Is it a relapse of the same set of bacteria after some time? Or is it a reinfection with a completely new set of players? We're also not in line with how to treat asymptomatic BV. Clinical guidelines still focus on treating only symptomatic BV, but there is evidence in papers that asymptomatic BV is also linked to inflammation, and there is no unified approach for a critical pathway to FDA approval for LBPs at this point. And I turn it over to Jacques.
Did I share the right thing? I don't think so.
No.
Sorry about this. This should be it.
Yes, and now just the whole screen. Yeah.
Okay.
Yes.
So what I'm going to do, following what Anke has just presented, is delve a little more on the what the vaginal microbiome is and some of its function, and later on we'll discuss how we can leverage this to try to innovate in live biotherapeutic development. So I have some disclosure I mandated to put out. So like Anke said, in reproductive age 1, this high abundance of lactobacillus is what we characterize as optimal for the vaginal microbiota. And just a point to note that it is a feature that's very unique to human, as no other animal actually carry lactobacillus in their vaginal microbiota in such a high abundance, and which makes studying this in a lab actually quite a lot more challenging than compared to the gut microbiome, for example. And like Anke said, lactobacillus produce lactic acid and can acidify the vagina pretty high, and we believe this is a way to restrict the growth of those non-indigenous microbes that cause BV, for example. However, this is not the only role of lactic acid, and this is why lactobacillus is also a very important species. It does produce lactic acid, but lactic acid does a lot more. And there's been a lot of papers that have shown that it is not only anti-inflammatory, so it has very good anti-inflammatory property, but it also enhances epithelial barrier integrity in the vagina and the cervix as well. And we've also showed that it actually helps also fighting pathogen by modifying the property of mucus, for example. And this actually, in this particular paper, we showed that D-lactic acid was actually very potent in modifying mucus structure to trap HIV in the mucus. So it made HIV basically immobile in mucus. When lactic acid was not there, HIV was able to penetrate mucus and ultimately potentially reach its target cells. So the vaginal microbiota, when we think about its structure, we published this almost like over 10 years ago, but we just revamped it recently by adding a lot more sample and data to this structure. And it was originally described as 5 different types of vaginal microbiota that exist in women. 4 of those are called optimal, and they're dominated by lactobacillus. And you have Lactobacillus crispatus, which is thought to be the optimal, the most optimal, if you want, lactobacillus. You have Lactobacillus cineres, which has been shown to be somewhat suboptimal potentially, and Lactobacillus gasseri, as well as Lactobacillus janseni. But there's a 5th larger group that doesn't contain lactobacillus, and in many ways it's very reminiscent of bacteriovaginosis, but in many women it doesn't present with symptom. So, which is a big conundrum in this field, is what to do with all those microbiota. And so from the original 5 community state type that we call them, we've been able now, with the large number of sample of over 13,000, we've been able to actually subdivide, if you want, those original CST and sub-CST. And now we have a much higher resolution, especially in that CST4 that lack lactobacillus. We have 7 of them that basically subdivide that CST4. So somewhat it can improve a lot of the study that we do with the microbiome. And from a longitudinal standpoint, that's the question I was always asked, because those 13,000 samples are cross-sectional. So from a longitudinal perspective, there's optimality is basically defined by a dominance of lactobacillus over time. And that dominance, in some cases, so in green here is dominated by Lactobacillus gasseri, in brown by Jensenii, in orange by Lactobacillus iners, in red by Lactobacillus crispatus. And you can see that even during episode of menses, there are some community that basically resist to change, others they don't, but they also change differentially. So, for example, this one has almost like a BV-like state, even though there's none of those prototypical bacteria like Gardnerella and Atopobium are not found in this particular community. This is Streptococcus, and here is Lactobacillus iners. And of course, there are a lot of women that have lactobacillus that persist over time, and you can see that even their response during menses could vary quite differently. So this one is iners, this one is another one of those community that looks like a BV but doesn't have those organisms. And here there's Streptococcus, Staphylococcus, and Enterococcus that actually can appear during menses. But as soon as menses is over, you can see that the community restore to its dominance by Lactobacillus crispatus. And this is a property that's actually really good, because even though you have a physical biochemical environmental disturbance of the microbiota during menses, as soon as those disturbance goes away, the restoration of a lactobacillus dominated state happens, meaning that it's a very resilient type of microbiota. And this is the kind of property we would like to be able to restore in women through the use of live biotherapeutic. And of course, we have what we call non-optimal, and you can imagine what they look like. They look BV-like type microbiota. And when we did that study, actually, it was quite surprising to see this state actually last for so long. Here we have 10 weeks, and you can see that this particular woman doesn't even have lactobacillus at all. So think about, treating this with antibiotic. If there's no lactobacillus, what is going to come back? So there's a lot of issue and potentially a reasoning for why you would want to use a live therapeutic that contain lactobacillus. So those very unstable that also are disrupted by menses. So to study the vaginal microbiome, we've developed a new tool that's we call that it's an analytical platform, but it at its core it comprises of a gene catalog of all the basically all the genes, the bacterial gene, fungal gene, parasitic gene that are found in the vaginal microbiome in a large number of women. So it's quite a comprehensive gene catalog. I don't know if you're all familiar with the concept of pangenome, but a pangenome, it's all the genes that exist in a species of a bacteria. So in different strains, you all combine, unique gene. So now we have the metapangenome, because it's all the genes from all the pangenome for each species. So the platform is called Virgo, and we use it to study the vaginal microbiome from a more functional perspective. We have a new version of Virgo that's going to be released shortly, and this one is a lot more extensive in its geographic representation, and the gene catalog now is about twice its size. It was derived from 2,500 metagenome, and over 15,000 genomes are in our databases. So the advantage of this catalog is that we have a very high level of taxonomic annotation. So we know the species that encodes the gene. Okay, so we have 92% of those that are taxonomically annotated, but we also have very high rates of functional annotation. About 85% of all the genes actually have some level of annotation. And we also have, as part of the catalog, on top of fungal and parasitic gene, we also have phage and viral genes are present also in a catalog. So we can use this catalog to perform what we call metagenomic and metatranscriptomics. So metagenomics is basically looking at all the DNA that's present in a microbiome. So we will be looking at not only just the bacteria, but also, so not only the taxonomy, but also the functional profile of the microbiome. But more importantly, because of those annotation, we can look at functional profiles stratified by taxonomy. And that's the advantage of using the catalog. And transcriptomics doesn't just look at the genes that are present, it looks at the genes that are being expressed by the microbiome at the same time. And so this is what I'm going to show you about. It's just a little snippet of what we've learned by analyzing a large number of metagenome and transcriptome that were collected over time in women. And we analyzed this with Virgo. So the first thing we learn is that actually the composition of the vaginal microbiome does not always reflect the activity of the vaginal microbiome. So what you can see here is the relative abundance, so the composition in proportion of each of the bacteria. So in red is Lactobacillus crispatus, for example, and in this teal is Gardnerella vaginalis. So here you have the lactobacillus dominated community, and those are like the BV-like community. And what you can see here on the bottom is the contribution to the metatranscriptome of each of those different organisms. So what the main message here is like, if you just look in the CST4 type community, you can see that Gardnerella, even though it's present in very high abundance, it's not always the one that's contributing a lot to the function of the community. Other organisms that are present in much smaller abundance actually are contributing a lot to the community. So, which means the looking at the function, at the genes that are being expressed is going to tell you a lot more about what's going on in the vaginal microbiome at any given time. So what we've learned from doing this is that, I'm just going to give you two little snippets. One of them is glycogen degradation. So it was thought for a long time that actually lactobacillus was not able to use glycogen. Well, it turns out that they are, and we've known this now for a little while. This is nothing novel. But what we show here is that glycogen is actually a property that is encoded in almost all the microbiome that we've looked so far. And whether it's crispatus that express the enzymes that are able to degrade glycogen, or Lactobacillus cinereus in orange, or even Gardnerella and Atopobium and Prevotula, they all will be able to use this. So glycogen is actually what I call a universal resource. The other one that's more important for the pathogenic property of certain microbiome is the expression of sialidase and fucosidase, for example, which would be involved in the degradation of mucus, which is evidently a very important component of the protective machinery in a vagina. And so what we see here is that the genes are basically only encoded and expressed by non-lactobacillus species. So you can find their expression in almost every CST. So whether it's CST1 dominated by crispatus, or CST3 dominated by iners, or CST4, but it's always those non-lactobacillus that do express them. But of course, there's a lot less expression in CST1 compared to CST4. So what this means is that lactobacillus does not use mucin or mucus as a source of nutrients, but this is something that is unique for bacteria like Gardnerella, Prevotula, or Propionibacterium that are often found in bacterial vaginosis. The other thing that's also very important is asking the question, those express function, are they modulated by other species? So basically what we're trying to say is that if you have, let's say, Lactobacillus cinereus in presence of other lactobacillus, is it expressing different gene compared to if Lactobacillus cinereus is cohabiting the vagina with Gardnerella, for example? And so we did this sparse canonical analysis of metatranscriptome and metagenomic data. And what we found is that, yes, genes are for each species are modulated by the composition of the community. So here for Lactobacillus cinereus gene, you can see that when it's in the presence of Lactobacillus jenseni, it's expressing a set of gene, and when it's present with anaerobes, for example, it's expressing a different set of gene, including mucin binding protein as well as cytolysin and neurolysin, which is very similar to vaginal lysin in Gardnerella, so a cytotoxin. And in Gonarella, you can see exactly the same thing. When it's present with Prevotella or Megasphaera, it expresses one set of gene, including vaginal lysine, but it's present with Lactobacillus, it expresses other kind of gene. So context is very important. And bacterial gene expression is totally modulated by that microbial environment. And in the vagina, what this study has found is that glycogen is a universal resource that can be used by any bacteria in the vagina. So ultimately, what we'd like to know is this optimal, non-optimal. And this is a slide that was originally put together by Caroline Mitchell. And what it shows here is that it's all a bunch of study around the world where they looked at the vaginal microbiome and looked at different outcomes, whether it was HIV, HPV, preterm birth, or even inflammation with IL-1 beta. And what they showed, all those studies have basically concluding is that lactobacillus is almost universally optimal, especially lactobacillus crispatus. And so this is actually was very well demonstrated by a paper, which I think this figure has already been shown, and I think Doug might show it again, where Doug, because it comes from Doug's group, where he followed a cohort of young South African women that were HIV negative and looked for acquisition. And what he found is that even though we need to, I know he has more, most of all the women that were originally in Lactobacillus crispatus dominated actually never acquired HIV, while the other, in other CST, including Lactobacillus inners, actually did. So the role of Lactobacillus crispatus in optimality in a vagina is definitely key. So basically, we can conclude that positive outcomes are always associated with lactobacillus dominance. And in the next slide, which I'll present in the next time I come up, is I'll tell you more about how do we leverage this knowledge and how do we restore lactobacillus dominance. Doug?
Great, thanks, Jacques. So I'll go ahead and continue the story. And since part of the focus here is the role of LBPs in HIV prevention, I thought I'd talk a little bit about sort of some of the work we've been doing about the interaction between microbiota, genital inflammation, and then the HIV risk. So just to kind of frame, HIV and sort of why we particularly focused on young women in sub-Saharan Africa, most new acquisitions are occurring within sub-Saharan Africa, and most of those acquisitions are occurring in young women of reproductive age. And you can see that there's this very disproportionate burden of infection in women in many of these countries in sub-Saharan Africa, where prevalence rates are, up to 7 times higher in some of these countries amongst young women than young men. And so we've been working for many years now with this cohort called the FRESH study. So FRESH stands for Females Rising Through Education, Support, and Health. And this study has enrolled over 3,000 young HIV uninfected women in this township just outside of Durban, South Africa, called Mlazi. They come to this site twice a week and they get tested for HIV using a finger prick PCR test. They also have routine pelvic exams and blood draws done every 3 months. They get tested for STIs, as well as they have pap smears done. And they're also involved in this poverty alleviation program where they spend, this each time they visit twice a week for 10 months, they're spending 3 hours there to learn job skills, learn how to use computers, use how to write resumes. And we're very proud of the fact that most of them go on after graduation to either jobs, internships, or other types of kind of meaningful activities. And yet the HIV acquisition rates within the group remain indistinguishable from those within the community and just really enormously high. So to give you a better sense of that, within sub-Saharan Africa, South Africa in particular has very high incidence and prevalence rates. And this region of KwaZulu-Natal, where the study takes place, has some of the highest rates within South Africa. So in this Vukuzazi study, which was done within a region within KZN, they went door to door and they tested almost half of the people living in this catchment area, so about 20,000 people. And what they saw was that in young women in their late teens, early 20s, and then all the way into their 30s, HIV prevalence rates exceed 60%. So that period, in the late teens, early 20s is a period just of enormous vulnerability for HIV acquisition. But there's some things that we know about the biology of transmission that make us hopeful that interventions can be created to reduce this acquisition risk in young women. And one of the main things is that we know that women, when they're exposed to HIV across the mucosal tissues within the female genital tract, they're sort of exposed to enormously diverse quasi-species of viruses. But that in most cases, it's a single virion that's able to infect what's thought to be a local activated CD4 T cell within the genital tract. And then there's this period of sort of local obligate expansion until you have enough viral burden to then disseminate, first probably to the draining lymph node and then very rapidly throughout the whole body. And that sort of local period of local dissemination is thought to take about a week. And that's been referred to as a window of opportunity, because if you could stop HIV there, you might actually be able to prevent this sort of dissemination and establishment of infection. And so, as has been mentioned, we kind of looked at the vaginal kind of microbial communities within women and fresh. And what we showed in kind of contrast to sort of what you see within women, for example, white women in the US, where most 90% of women will have dominance by a single species of Lactobacillus crispatus. In our case, most of the women, about 2/3 of them, actually had dominance by these mixed anaerobic communities with low Lactobacillus crispatus, with low lactobacillus abundance, which Jacques has called CST4. And then only a minority of women, about 10%, actually had what's thought to be this optimal community that Jacques and Anke had kind of talked about, which is a Lactobacillus crispatus dominant community. So we then showed that these communities, which we called CTs, actually very closely correlated with different measures of genital inflammation. So if you looked at inflammatory cytokines within genital secretions, you could see that women with Lactobacillus crispatus dominant communities, or CT1 in this figure, had much lower levels of inflammatory cytokines than women with these CST4 mixed inflammatory communities. And that was looking at all cytokines in aggregate, or actually looking at individual cytokines across over a dozen different cytokines. There are also increased measures of activated CD4 T cells within the genital tract in women who had these CST4 type communities. And you could see 17 times more activated tissue resident CD4 T cells within the cervix in women who had this mixed anaerobe dominated community than women who had Lactobacillus crispatus. And since I mentioned that these are thought to be the first cells to be infected by HIV, it's thought that may be one of the mechanisms by which vaginal bacteria can increase biological risk of acquiring HIV. And as has been kind of shown, as been discussed, we then showed that these women also acquired HIV at over 4-fold higher rates than women with Lactobacillus crispatus dominant communities. What's interesting about sort of the inflammatory signals within these cells is that if you kind of look at different cell types, antigen-presenting cells have the most differentially expressed genes when you look at women with Lactobacillus crispatus dominant communities and these CST4 type communities. And one of the pathways that's most differentially regulated is LPS sensing, or TLR4, which made sense because if you look at the bacterial communities, these CST4 type communities actually have a lot more LPS with them, or lipopolysaccharide, than Lactobacillus crispatus communities. But what's also interesting is that a lot of these inflammatory signals that we see in women with these CST4 inflammatory communities actually also match the same kind of inflammatory signals that had been described from much older literature in, say, preterm birth, where things like upregulation of IL-1 beta and TNF, upregulation of LPS signaling, NF-kappa B, other cytokines like IL-8 had been described as being associated with preterm birth, but not necessarily in the context of a specific type of vaginal microbial community. And so there's a lot of overlap between these. And I think that, now there's a greater appreciation that, in fact, these things may be related. And that, in general, having a lactobacillus dominant vaginal microbiota is associated with having more positive outcomes for a number of different sort of reproductive outcomes. So that includes decreased rates of preterm birth, lower rates of tubal infertility, lower rates of pregnancy loss, lower rates of cervical dysplasia and cervical cancer, and better success with in vitro fertilization, and then lower rates of other STIs besides HIV. And I think importantly, many of these different adverse outcomes have a much higher burden within low and middle income countries. So I think that, a lot of us feel that, having a way to more effectively alter and durably change the vaginal microbiome may have an impact on reducing a number of these really important adverse outcomes. Now, the problem with standard of care treatment for vaginal dysbiosis, or in this case, bacterial vaginosis, with the antibiotic metronidazole, is that one, it's not really effective at actually achieving this more optimal Lactobacillus crispatus dominant community. What you see is that, and this has been known for a while, is that when you treat all these anaerobes with metronidazole, what you end up getting is Lactobacillus inners dominant community. And one of the reasons why we think that that could be challenging is that when we look at longitudinal data within women in FRESH, that when there are transitions between Lactobacillus crispatus and these CST4 anaerobe dominated communities, what happens is that they generally transition through a Lactobacillus inners intermediate. So Lactobacillus inners, at least within this cohort, seems to be a promiscuous state that sort of facilitates transitions for more optimal Lactobacillus crispatus dominant states to these sort of more inflammatory disease-associated anaerobe dominated states. And then once you're in these sort of CST4 type communities, what happens is that you can kind of see there are these short sort of path links between them, so that they're very adherent, and that you tend to kind of stay within those communities. So optimally, in a perfect world, what you would have is you would have interventions that could actually kind of get you out of this vicious circle of like having diverse anaerobes and then getting treated, and that gets you Lactobacillus inners, which actually provides high probability of recurrence to Lactobacillus inners. And what you'd want is to have an intervention that could actually drive your community all the way to a Lactobacillus crispatus dominant state. But that doesn't really exist right now. And so we were interested in kind of looking at various metabolites that actually may be important for kind of modulating these vaginal bacterial communities. And we are particularly interested in unsaturated long-chain fatty acids. And that's because it's known that these long-chain fatty acids are rich at mucosal surfaces, and they've been described as being sort of sensed and utilized by a lot of different bacteria, and also in certain cases can actually inhibit bacteria. So palmitoleic acid, for example, has been described on the skin as being an inhibitory compound for Staph aureus growth. And so we looked at a variety of different unsaturated long-chain fatty acids. And in particular, we were really interested in oleic acid because in our hands, it had this property where it seemed to be able to inhibit the growth of Lactobacillus inners here, but not necessarily other lactobacilli. And then actually, if you put it under different media conditions that were optimized for Lactobacillus, non-inners Lactobacillus growth, what you could see that actually oleic acid also had this property where it could actually promote the growth of these non-inners lactobacilli, but not Lactobacillus inners. And so that seemed like a kind of, and then when you looked at Lactobacillus inners specifically, oleic acid seemed to have this property where it would actually kill Lactobacillus inners, but not these other non-inners lactobacilli. So we wanted to kind of better understand what properties of oleic acid and some of the other related unsaturated long-chain fatty acids might actually be conferring this. And so we looked transcriptionally at bacterial activities after being exposed to oleic acid. And what we saw was that there's actually a conserved genetic response to oleic acid between multiple different non-inners lactobacilli. So, and that consists of three genes primarily. One is OHYA, which is a enzyme that hydrolyzes oleic acid. Then there's FAR-E, which is a pump that transports oleic acid, and then TET-R is a regulator of that that transporter. And when we looked at different genomes between different lactobacilli, what we saw was that Lactobacillus inners uniquely lacked those genes, the OHYA and the FAR-E genes, whereas other non-inners lactobacilli actually almost uniformly had those genes present. And in this work, what we showed was that, in fact, non-inners lactobacilli can actually take unsaturated long-chain fatty acids like oleic acid and can use FAR-E to import them, and then actually utilize them to actually form phospholipids, which are then used for cell growth. It can also interestingly use OHYA to hydrolyze oleic acid and other unsaturated long-chain fatty acids, and then basically store this metabolic state in a sequestered form that prevents it being utilized by other lactobacilli that don't have this OHYA gene. So we found that really interesting. We demonstrated that these genes were important by, for example, knocking out OHYA and showing that if you took Lactobacillus gasseri and you knocked out OHYA, that it actually caused Lactobacillus gasseri to now die. And then if you complemented back, then it could actually then still use oleic acid as an energy source. And then we thought, well, could this actually be leveraged to then try to affect vaginal microbial communities? And so what we showed was that if you take oleic acid alone, that'll increase Lactobacillus crispatus, either alone or in combination with metronidazole. But interesting, and then of course, it'll decrease Lactobacillus inners. But interesting, oleic acid alone actually could also just by itself inhibit some of these anaerobes that are present in these BV-like communities. And so we kind of modeled this by creating these sort of mock mixed communities where you could see that there are sort of different anaerobes mixed with different lactobacilli, sort of replicating kind of BV. And if you treated with metronidazole, what you got was Lactobacillus inners dominance, which is sort of mimics what has been observed, within people. But if you treated with oleic acid or even its metabolite 10-HSA, its oxidized metabolite, you could actually promote Lactobacillus crispatus dominant communities. And if you did that in combination with oleic acid or 10-HA and metronidazole, you could actually do an even better job of promoting Lactobacillus crispatus dominance. So we're actually hopeful that actually might inform not just the biology of vaginal microbiota, but also things like potential future interventions. And so, I kind of talked about in the context of HIV, Lactobacillus crispatus is associated with this low inflammatory state within genital tissues. But when those communities are replaced by the CST4 anaerobe dominant communities, you have a lot of inflammation there. That results in increased activated CD4 T cells and increased HIV acquisition risk. But there are probably also a lot of other adverse effects of these anaerobe dominating communities that sort of degrade the barrier function within these mucosal tissues of the female genital tract. And so in my next segment, I'll talk a little bit more about like some of the data that we've kind of been trying to generate, looking at different approaches to changing the vaginal microbiome. And I'll stop there. And so I think back to Anke now.
Yes, replace current share. All right. So in this next round, we will talk about the various trials that we have implemented and analyzed. I will start and then turn it back over to Doug. Together with Ocell, a biotech company here in California, and Craig Cohen at UCSF, we've been working for 15 years trying to develop live biotherapeutic products, initially for the prevention of bacterial vaginosis, in a product with a Lactobacillus crispatus strain that is endogenous to the human vagina and called Lactin V. And our hope is that by decreasing the risk of BV recurrence, that we can also impact all these other conditions. An LBP has a clear advantage. It is female controlled. It could eventually be low cost. It is non-coitally dependent, so women can take that at any time. It's not related to sex itself. And it could be self-replicable in situ, so you have to do only very intermittent dosing. We are also working on what we're calling a second generation LBP that is a genetically modified lactobacillus strain that can express an antiviral drug compound. And I'll talk about this a little bit later. But I'll start with our latest LACT-V study in the US. We try to really learn from studies that came before us and do a study design that is really robust and tries to avoid all those previously made mistakes. So very on the study product side, very frequent dosing in a vaginal applicator, giving the LBP pretty much right after the antibiotic course is finished, doing long-term colonization during dosing, but also having a post-dosing phase, trying to detect our specific lactobacillus strain that is in LACT-V, and also collect a whole number of important data in the daily diary on adherence, semen exposure, menstruation timing, contraception, vaginal practices, all of those things, so we could link that to the outcomes. And we try to do really robust BV assessment following the 2013 FDA guidance at the time. And we were very fortunate that in a later stage, the Gates Foundation, through Doug Kwon's lab, funded the secondary analysis with molecular methods. And Doug will talk about this in a minute. So we enrolled 228 women across 4 sites in the US, randomized them 2-to-1 right after they finished their Metrogel treatment, and then gave them product over 11 weeks. And the main outcomes were at 12 weeks right after the product ended, and then another 12 weeks later at 24 weeks, measuring colonization, side effects, and recurrence of BV. So very straightforward study outcomes centered around efficacy and safety and colonization at 24 and 12 weeks, and also acceptability and tolerability. And we were very excited to see when we finished analysis in 2019 that indeed LACT-V was preventing BV recurrence significantly in comparison to placebo. But this effect also prevailed to a lesser extent, but prevailed even 12 weeks after product stop. So, however, the 30% recurrence in the LACT-V group still is pretty high. Part of it could be that this was a cohort that was very highly motivated because they had a long history of BV. So these were very complicated cases. What we also looked at and were really excited to see here, the gray bars, that throughout the dosing phase, as well as 12 weeks later, colonization reached 80% in the LACT-V group, and even at 24 weeks, 50%. And among those women that did colonize with CTV-05, the level of the concentration of CTV-05 was really very stable throughout, even after product had long stopped. So that was another very exciting result. But as I said before, those 30% recurrence, even in the LACT-V group, was something we really wanted to study in more detail. So we did, with the data that we already had collected, a post-hoc analysis to basically stratify those women that had already achieved clinical BV cure per AMSL. So there were AMSL 0 right after they came off metronidazole. We found out, we were actually surprised that this happens really fast. Within 48 hours after metronidazole, 88% of women where AMSL 0 achieved clinical cure, and there were only 12% that did not. And when we stratified that and then looked here and compared in blue the BV recurrence rates among the women that were cured after metronidazole here, 25%, and the one that were not cured, you can see a significant difference. We also, not pictured here, looked at colonization levels that were significantly higher in the cured group after metronidazole, and they also reached higher levels of CTV-05. And this effect also, a little bit less, but also prevailed at 24 weeks. So this leads us to believe that the initial antibiotics really clear the niche for LBPs and can really contribute to the success of the colonization of an LBP product. Before Gates funding arrived, we were trying to do a smaller study, taking the 66 participants where we had complete sample series for all 7 time points from baseline to 24 weeks. And Eric Armstrong and Rupert Carlslath at the University of Toronto published extensively on this. And I'm just going to summarize this very basically, that it showed that those colonizers had an increased overall and strain-specific lactobacilli load, and also the markers for epithelial disruption decreased, while we did not see that in the non-CTV colonizers. And he was also able to show that the initial Metrogel reduces, not just clears the microbial, the pathogens, but also reduces inflammation. All right, and I will turn it over to Doug who gives you a little more information on the metagenomic time course data that we did with our 2B samples.
Great. Okay. So yeah, so to talk a little bit more about, as Anke mentioned, the molecular analysis of the 2B trial, and then also some results that we've done with the Lactin-V product in FRESH, the cohort in South Africa. And then I'll end with just sort of some direct comparisons. So, as Anke mentioned, this was sort of the 2B trial that was published in the New England Journal in 2020. And here the endpoints were mostly kind of clinical, which were sort of based on clinical measures of recurrent BV. So we used 16S sequencing to kind of look at sort of at a kind of taxonomic level, sort of what were the results. And what we saw was that overall there was definitely much more Lactobacillus crispatus, which is here in orange, in women who had Lactin-V, which is sort of here on the top, versus those who were in the placebo group. And if you looked at that sort of in a Sankey chart, what you can see is that this is when they, this is 4 weeks into treatment, and then this is when they stop treatment, that you can see a lot more Lactobacillus crispatus here in orange in women who received Lactin-V than women who just received antibiotics alone. And what that looked like in terms of a benefit ratio is that essentially at week 12, there were 3 times more women with at least 50% Lactobacillus crispatus within their vaginal microbiome in the Lactin-V group versus placebo. And then at 24 weeks, which is 12 weeks after stopping delivery of the product, actually that was seemed to persist fairly well. Now, one question that you can do by doing a molecular analysis is kind of look at actually strains of different Lactobacillus crispatus. So here, rather than doing 16S, bacterial 16S amplicon sequencing, we actually did shotgun metagenomics here. And what we saw is that if you just kind of look at the women who had at least 5% Lactobacillus crispatus, and here, so women who didn't have Lactobacillus dominance are basically, or Lactobacillus crispatus were here in gray. And then here, what you can see is that those women who had CTVO5 are here in darker orange. And then women who had another strain, mostly another strain of Lactobacillus crispatus, are here in yellow. And what you can see is that at week 4, which is 4 weeks into treatment with Lactin-V, that most women who had Lactobacillus crispatus had CTVO5, the strain of Lactobacillus crispatus in the LBP. But at the end of treatment at week 12, there were actually some women, in this case, 15 women who actually had dominance by a different strain of Lactobacillus crispatus. And that was kind of maintained even after 12 weeks after stopping. So if you kind of look at sort of how Lactobacillus crispatus changed over time. So if you look at kind of starting points where these are women who have Lactobacillus crispatus, and almost all of their Lactobacillus crispatus is CTVO5. These are women at time points where they have a mix of CTVO5 in orange and then some other strain of Lactobacillus crispatus in yellow. And these are women who have mostly non-CTVO5 Lactobacillus crispatus. And then you look at sort of the next time point, what you can see is that most of the women who had CTVO5, actually, they transitioned to a state where they kept CTVO5. Some of them transitioned so that they no longer had Lactobacillus crispatus dominans. And then there were a small number of women who went from having mostly CTVO5 to actually then having mostly a different strain. Those women who didn't have Lactobacillus crispatus mostly continued to not have Lactobacillus crispatus, although some did acquire it, and some acquired CTVO5 and some acquired a non-CTVO5 strain. What's interesting is that there were, a handful of women who actually had mixtures of CTVO5 and non-CTVO5 Lactobacillus crispatus in more equal proportions. And what you saw was that at the next time point, that most of these women seemed to actually have the non-CTVO5 strain kind of win out, although some of them did have the CTVO5 strain win. So it was interesting, kind of in terms of the strain dynamics, what it seemed is that like most women who established Lactobacillus crispatus colonization had that with CTVO5, but in women who had a mixture of CTVO5 and a non-CTVO5 Lactobacillus crispatus strain, where there seemed to be some competition, that endogenous strain seemed to sort of win out, even though that was a minority of the participants. We also kind of did a kind of analysis to kind of look at what are the factors that are associated with successful Lactobacillus crispatus colonization. So these kind of arrows represent different factors that were found to be significant. And so the length of the arrow kind of tells you sort of the weighting of that factor. And it's overlaid on a background of sort of these communities. So this axis 1 mostly discriminates between lactobacilli and non-lactobacilli. And this axis 2 mostly discriminates between lactobacillus inners and Lactobacillus crispatus. And so what you see here is that if you look at post-metronidazole bacterial load, that was actually one of the strongest factors that predicted Lactobacillus crispatus colonization at week 12. Other things were alpha diversity and pH, so that if you had a higher alpha diversity, you were less likely to get Lactobacillus crispatus dominance, and an elevated pH, which basically meant that you actually just didn't, you basically post-metronidazole, you continue to have an anaerobe dominated community. We also saw that pre-metronidazole, so even before you took antibiotics, alpha diversity actually and community composition actually had some effect on some ability to predict outcomes, so that women who had higher alpha diversity before actually were less likely to colonize with Lactobacillus crispatus. And then interesting, there were certain cytokine signals that seemed to negatively or positively correlate with Lactobacillus crispatus colonization. Again, this is pre-metronidazole. And sort of what that means, it's not entirely clear, but if you were going to speculate, you might think that, oh, women who had a stronger inflammatory response to these BV-like communities seem to be a little bit more likely to colonize with Lactobacillus crispatus. And so maybe in those cases, the kind of mucosal responses within the genital tract may be sort of helping with Lactobacillus crispatus colonization, but that's entirely speculative. And then there are some demographic factors. So declared race was something that actually also associated with successful colonization. Oh, and then bleeding. So bleeding between after metronidazole, the week 4, women who reported more bleeding were less likely to colonize with Lactobacillus crispatus and more likely to colonize with actually Lactobacillus inners. So just very briefly, we also had the opportunity to do a smaller trial in FRESH, which is in South Africa again, of Lactobacillus crispatus, of Lactin-V. And here I'm just kind of showing you a comparison of the 2 different trials in the US, the 2B trial in the US and the trial within FRESH. So there were, certainly differences. First of all, there was this difference of different populations. There was also a difference in the metronidazole that was provided up front. So in the US, it was vaginal metronidazole that was given for 5 days. In South Africa, it was oral metronidazole that was given for 7 days. And then there was the duration of treatment, where in the US, women were treated for 11 weeks, and in South Africa, they were treated for 4 weeks. But there are some kind of similar kind of follow-up time points. And so if you kind of look at, and also the size of this study was sort of different. Yes, and these are just sort of listing kind of some of those differences I went through. So if you kind of walk through kind of what it looked like looking side by side at North America and South Africa. So at screening, again, women mostly had these mixed, diverse anaerobe dominant communities. Then post-metronidazole, most women shifted to a lactobacillus inners dominant community here in green. And then if you look just at the point of product cessation, and again, as a reminder, that was week 12 in the US Lactin-V study, or week 4 in the South African Lactin-V study, you can see that in general, there's more Lactobacillus crispatus established in both studies within these populations with Lactin-V treatment. But what's interesting, if you look 12 weeks after, the response seems to be more durable within participants in North America than women in South Africa. And then there were some other kind of intermediate time points that don't quite match up, but that's kind of how those time points look like overall between the two studies. So if you kind of look at this benefit ratio, again, in the US Lactin-V study at week 12 at product cessation, 3 times more women had Lactobacillus crispatus dominance in the treatment arm than in the placebo arm. And that pretty much persisted even at 12 weeks after stopping. In South Africa, you had a similar result, and possibly even a little bit better, where after 4 weeks, 5 times more women in the Lactin-V arm had Lactobacillus crispatus dominance than in the placebo arm. But then what you saw was that they seemed to actually lose this, that it wasn't as durable. And why that is, obviously, we don't know, because there were a number of important differences between the two studies. And so whether or not that had to do with, the way metronidazole was given, the duration of treatment with Lactin-V, or the differences in the populations, I think remains to be seen. So I'll kind of stop there and just thank, in particular, some people that were involved in this Lactin-V analysis, which include Craig and Anke, importantly, but also Laura Simel and then Seth Bloom. And then just for the first part of the talk, just thanking some of the people in the group who kind of were involved in those studies. So with that, I will stop, and then I will pass it off to Jacques.
Okay, so last time we stopped is how do we restore, and we got to hear quite a very nice story about initiatives that are ongoing on trying to do this. And so the product that have been presented have been basically contain one strain of the live Biotherapeutics. And we have another initiative which Doug is part of, where we're developing a multi-strain live biotherapeutic product. So it's a single species, but multiple strain of that species. It's funded by the Bill and Melinda, well, I guess now it's called the Gates Foundation, under a project that's called the Vaginal Microbiome Research Consortium, or VMRC. And it's a group, large group, actually, there's some missing here, a large group of scientists from around the world, in the US, in Africa, in Bangladesh, and so on, that are all working to develop an innovative live biotherapeutic product for bacterial vaginosis and ultimately potentially just CST4, to reduce the risk of reproductive and gynecological adverse outcome in Africa and South Asia. So the big question is, why a multi-strain formulation? And this is where we're trying to learn from ecology. So in analyzing this gene catalog called Virgo, and analyzing metagenomic data set, what we found is that if you count the number of non-redundant genes that belong to a specific species in a metagenome compared to that of a single strain genome, what you find is that in a metagenome, you have a lot more gene that belong to that species than what's encoded in one genome. So Lactobacillus crispatus is about 2,000 gene on its genome. And so if you have one strain of crispatus, you should have about 2,000 gene. But in reality, we have a bit over 3,000 gene, which means that in the context of the pangenome, that means there's multiple strain of that species that coexist, each bringing genes that are not in the others. And so this is what we call an assemblage of conspecific genotype, meaning strain of the same species that exist in vaginal microbiome. So the idea here is that if you have one genome, one strain, then if you look at all those genes that are present in that metagenome, meaning that in the ecology that's happening at the time in the vagina, in that microbiome, you're only representing about 60%, maybe 50% of the entire set of functions that are present in that environment, in that metagenome. So what we wanted to do is to basically use the information to design a live biotherapeutic product that comprise multi-conspecific genotype of Lactobacillus crispatus in order to reproduce all the functions that are present in a metagenome, reproduce them as much as possible into our live biotherapeutic product. So if you think about it, the way you can think about it is that those conspecific genotypes, so those strains of the same species, when they assemble together in a vaginal microbiome, basically they're going to be differing in their genetic content. So what you can see here is that those different assemblages. So if in one woman you have 20 species, that 20 strain or 10 strain that are together, those 10 strain are going to have a set of function that could be different from the 10 or 15 strains that are found in another woman. And so when you start looking at this, what you can see is that it's true. You can actually cluster those assemblages by their genes. So here, this is an example, and it's true for a lot of different species. This is Lactobacillus cinereus, for example, and you can see that the set of genes that are present in those particular women, so each woman is a vertical line, is different than those that are present in this one. So you can start seeing those group of women that have assemblages of Lactobacillus cinereus that tend to resemble each other functionally. So they basically carry the same functionality. And so we think this is very important. And so we went on and, another thing that I want to say, another advantage of having a multi-strain consortia is that we know that when you have a one-strain formulation, it will colonize some people, but not others. Okay. And so one of the thought behind this is that if you have multiple strains, it's possible that some women will be colonized by all three strains, or maybe just two of them, or three, but in different proportion, or just one of them. And maximizing the number of women that potentially could be colonized by strains that are present in a formulation. So that's another thing. But this is how we actually build this LBP. So we're calling building a microbiota informed live biotherapeutic. So what we did, we have those pattern, and that was not those exact one, but we have women that are optimally colonized by Lactobacillus crispatus, and so we sequence their metagenome over time. So we have metagenome from all those women. So we have actually now a gene set for each one of those time points that we sequence. So it's all the genes are encoded on Lactobacillus crispatus in those communities that are dominated by Lactobacillus crispatus, okay? And then we have a large collection of Lactobacillus crispatus strains that have been sequenced that are genetically unique. And they all come from women who have very stable community. And we build a heuristic algorithm that takes the gene catalog, gene information from each metagenome, take the genome, so all the genes are encoded on those genome, and try to reproduce this gene that's found in a metagenome by assembling a whole bunch of genome together. And while minimizing the number of genome you want to be incorporated and maximizing the number of gene. So what happened is that we have the algorithm has generated several candidates of 6 Lactobacillus crispatus strains. This is where you get anywhere, between 80 and 90% of all the genes with 6. And also financially, it's also a little more responsible. So we now have several of those candidate consortia. One of the things that we also did, because we found that there are strains of Lactobacillus crispatus that are found basically worldwide. You find them everywhere. But there are strains that are very specific to certain geography. And so we tried, so you can see here, those 3 strains, for example, have only been found in Africa. Well, those 2 strains, one is from a US collection and the other one is in Africa. So there are strains that and lineages of Lactobacillus crispatus that are unique to geography and others that are shared across geography. So we wanted to reproduce and incorporate a lot of those strains. So we have strains, and also because the product is intended to be used in Africa and South Asia, so we wanted to use strains from the local geography for this first product. And so we took all those consortia and we screened them for their antimicrobial property against a large set of organism and multiple strain of those different organisms. And what we found is that VMRC1, that was originally called VMRC1, was always pretty much consistently the one that had the highest antimicrobial property across this wide range of organism. And that's the consortium that we took on, and it's been manufactured, and it's now called LC106. It's a 6 strains consortia. And we also have an experimental consortium, which we call LC115, 115, which contains 15 strains. And the idea here is to see this idea of colonization. Are there better colonizers than others? There are strains that colonize everywhere and so on that we might not have captured as part of our selection process. So we might learn a lot by doing this. We don't anticipate this to be a product, but I think it's experimental to try to learn about strain colonization. And so now we have a 1 gram mucoadhesive pill that has been packaged in blister, will be delivered with applicator. You can see it here. And we have designed a trial that is led by Caroline Mitchell at MGH, Doug Kwon also at the Ragon, that you know, and Disebo Potlan, who's at Caprisa. The trial is called Vibrant. It's vaginal live biotherapeutic randomized trial, and it's for the prevention of recurrence of bacterial vaginosis ultimately. And here's the design of the trial. There's 5 arms in the trial. Obviously a placebo. It's after oral metronidazole, and it's given 7 days daily. And the idea is to look for colonization over at 5 weeks as well as 12 weeks for extended period. The LC106 is given daily. We have also a shorter dosage of 3 days. And then we have another arm where we deliver the LBP at the same time as metronidazole. And this has also been supported by data that we've shown where if you expose Lactobacillus crispatus to metronidazole, there's actually transcriptionally, there's a 0 response. No genes are differentially expressed whether you have metronidazole or not. So this Lactobacillus crispatus is completely inert to the presence of metronidazole. So there's no reason to not deliver it at the same time as antibiotic. As we all know, and we've shown in the figures that have been shown, Lactobacillus iners tend to come back very early during the treatment. So it would be advantageous to have that high dose of Lactobacillus crispatus at the time or where the niche is opening ecologically for colonization of the LBP as opposed to Lactobacillus iners. So we hope to be able to observe this actually in vivo. And we obviously have the 115. So the trial will take place at 2 sites, one at MGH in Boston, that's led by Caroline, and one at Caprisa in Durban, South Africa, that's led by Disebo here. And the laboratory work for this trial will be done here at the University of Maryland School of Medicine, in Doug's lab at the Ragon, as well as at Caprisa, led by Sinai and Kapu. And with that, like Doug, I'd like to thank the people in my lab and the people who are part of the VMRC for this work. I think it's time for question.
I have one more round, just telling us what we are having in the pipeline. Just a couple of slides. So Lactin-V was also tested in a number of new indications here. BV for preterm birth prevention, phase 1 feasibility study was done in the UK that showed, compared to historical controls, Lactin V was able to lower PTB, and now the next step will be a phase 2 trial in London, together with the funded by the National Cancer Institute Cancer Prevention CT Clinical Trials Network. We are planning a study using Lactin V in women with high-risk HPV infection and vaginal dysbiosis, this time giving product up to 24 weeks, so longer than before, and following them over a year, and seeing if their HPV clearance can be improved. We are waiting for manufacturing of study product and hope to start this in 2025 in 3 sites in the US. And we just submitted our just-in-time documentation for UO1 grant, testing our first recombinant LBP mucocept in a first-in-human study. And what we're basically trying to see here, is it safe, does it colonize, and can we kill it? And that study will start in early 2025, we hope. And if that works, if we can genetically engineer lactobacilli to produce drugs or antibodies against HIV or other STIs in the future, that could really move the needle another round. So we're really hopeful for this first study, and are also thinking about how to use LBPs for multi-purpose prevention technology. Potentially, LBPs could be put in a pod ring. Ocrest has been working on that, and OCell is communicating with them to see if Lactin V could potentially be loaded in a silicon pod ring. So I also want to thank everyone on our end. Some of the names you will recognize, and I hope today was a good session, lots of learnings, lots of new information that we will all need to digest, and I think we can open it up for questions.
Wow, that was just really fascinating work that you all shared. Thank you so much, Anke, Jacques, and Doug. Yeah, we have just a few minutes for questions. Let's see, I am gonna share. Yeah, so if you all have any questions, why don't we just open it up and raise your virtual hand. And we can, and you can unmute yourselves for the last few minutes. And I just, as you're doing that, if anyone does have any questions, I just want to remind you that the webinar recordings and other resources will be on the IMPT website. We have a section called MPT Learning Events where the recording will be. And I see Laurel. Laurel, great to see you. You can unmute yourself.
Is there any evidence that a multi-strain product will colonize better given the context of lack of metronidazole treatment, or like a metronidazole treatment that's inadequate? Do you have any evidence, Chuck?
I'm not understanding the question.
Do you have any evidence that a
No.
multi-strain product will work better than a single strain product?
But what that I understand, but the metronidazole aspect of you.
When metronidazole fails to clear bacterial vaginosis.
I mean, you know, just like when you started with Lactin V, we don't know. That's why we're doing the trial, to look for colonization. I mean, we know that in vitro.
We don't have any data on that yet.
In vitro, they compete pretty well. They stay together, but you know, in vivo, I don't know what. You know, we'll see what we find.
Okay, great. I see quite a few questions coming in the chat and hands raised. So I don't know if Anke, Jacques, and Doug can stay on any extra time, I see Anke can. So, and if not, we can maybe send you the questions and we can get back to you, to those, to the group. Yeah, so I just want to thank.
There's one more
everyone
question, I
again.
think.
Yeah,
There's one.
yeah, mine is fairly quick, and you guys probably
Sure.
have already answered it. This is Lisa Say from Sister Love, and my question is that did you find any differences by age group with any of the study participants that was notable?
We did not, although the age, at least in fresh, the age range is pretty narrow, 18 to 23. I don't know, Anke, if you looked at that in the 2B.
Yeah, and I try to remember, it was in Laura Simel's you show in one of the graphs, but these were all premenopausal women, 18 to 45. I think we had an average age of around 30, so not that many women where we could suspect that they have a somewhat different vaginal microbiome because they're maybe closer to perimenopause. But I think that might be another frontier to think about what live biotherapeutics could do in a menopausal vaginal microbiome, but that is very different and not as dominated by lactobacilli, so we may need a different treatment approach there.
Thank you. Thanks.
And there's one, yeah, you see it, okay.
Yeah. No, go ahead. Yes, do you want to answer that, Anke? Or?
Is there a plan to include oleic acid with lactin V for treatment, following up to what Doug had presented? I think that's an interesting concept, but whenever you add another component to a drug, obviously another FDA pathway opens up. So I think doing things like this would have to be very designed very purposefully on the clinical level, but I don't know, Lactin, Doug, if you have anything to add, if anything could be done, in the lab or something on that end.
Yeah, I mean, I think we think that would actually be a productive path, like, oleic acid with a crispatus containing LBP. So we're kind of looking at options, but I think that's a good thought.
Any other questions? Yeah, I just want to thank you all again, Anke, Doug, and Jacques. Fantastic presentations. We'll be circulating the recording. And I just want to thank everyone who joined us today too for this discussion. So thank you, and hope you all have a great rest of your days.
Bye-bye.
Bye.
Bye.
Yeah, bye.
Bye.